ladies and gents, it's Miss Porter, and today I'm going to be talking about the new information that we're learning about, which is lactose tolerance versus lactose intolerance, and I'm going to connect it to the old information that we learned earlier in the year, things like enzymes and mutations, and kind of how all of this goes together. So if we look at this next slide here, we see that there are 4 different biomolecules. And remember that bio means life. So when we're looking at 4 biomolecules, those are the 4 main types of things that make up all living organisms. So those 4 biomolecules are carbohydrates, like known as carbs, nucleic acids, proteins, and lipids. And carbohydrates and also types of proteins known as certain enzymes are going to be what we focus on today. So carbohydrates are like sugars, anything that breaks down into a sugar. Nucleic acids would be DNA and RNA. So if you think back to that unit, we talked a lot about the differences between those. Proteins would be different types of structures that are made up of amino acids, and those have certain functions within the body. So we talked about like collagen and elastin and hemoglobin and all those types of proteins. And then the only thing we haven't talked about yet are lipids, and those are different types of fats. So we'll get to those later in the year. We're just not quite there yet. Now, if we're looking at carbohydrates, that's just another term for sugars. So again, carbohydrates always break down into sugars. And the term saccharide is kind of a more formal term for a sugar. And if you look at the pictures there, you should notice some words or some prefixes that seem real familiar to you. And those prefixes would be mono and poly. Mono means one, and then poly means many. So if you're thinking of like a monocle, that would be a one little eyeglass that someone would wear, or poly means many, like a polygon has many different sides. So monosaccharides are simple sugars, and then polysaccharides are more complex sugars. There's also a term, and I just want to introduce it to you, but I'm not going to use it a whole lot, and that term is a disaccharide, and that would be a more complex sugar that's made up of 2 simple sugars. So again, I'm just going to use the term monosaccharide, 1 part sugars, and polysaccharide, 2 or more part sugars today. So if you look at the table there, you should notice that glucose and galactose, that last piece there, this one right here, this should look real familiar to you because we've been looking at glucose and galactose and how those simple monosaccharides, those 1 part sugars, make up the more complex disaccharides or polysaccharides like lactose. Okay? So if you think about that, then you're kind of in the game here and getting ready for this next part. Now, if we look at digesting lactose with an enzyme, we know that enzymes have a lot of different functions, and those are enzymes are specific types of proteins, and they're important in living organisms because they speed up reactions. So if you think about all of the reactions like the metabolism, like breaking down different things within the body to use for energy, keeping everything clean, you know that those reactions have to happen really quickly and really efficiently. So enzymes speed up reactions, and how do they do that? Well, they lower the activation energy, or the energy needed to start a chemical reaction. So that's the function of an enzyme in review. Now, lactose is the primary sugar found in milk, and it is a polysaccharide made up of 2 sugar monomers, or monosaccharides, and those monosaccharides are galactose and glucose. So kind of the crazy thing about lactose is that it's such a large sugar that it cannot fit through the cell membrane. It can't go across the cell membrane and into our bloodstream. So what happens is we have to be able to break that lactose down into the monosaccharides, the galactose and the glucose, so that it can go through the cell membrane into the bloodstream so that we can get nutrition from that. So how does that happen? Well, we have to have an enzyme that's going to be able to break that down. So, as we talked about before, we know that lactase is the enzyme that breaks down lactose. And remember that enzymes usually end in an ASE, and the substrate that they act on, or the substrate that they fit in with in the active site, is usually ending in an OSE. So lactase breaks down lactose. So if you look at that diagram here, you can see that that lactase enzyme is kind of crazy looking. It has a really complex 3D structure. And the function of that protein, that enzyme, lactase, is determined by the different levels of folding of the amino acid chain. So that's kind of just a quick review of protein synthesis. So if we look at this diagram, we can see that lactose, the polysaccharide or the complex sugar, is broken down into two monosaccharides through a hydrolysis reaction. So basically what happens is that water is added in the presence of lactase, and remember, hydro means water and lysis means to split. So it literally splits the lactose into two smaller particles. So lactase is normally released in the small intestines, where this step of digestion would take place. Well, what happens if you eat lactose and you don't have any lactase? Well, that would be what we call lactose intolerance. So we know that this causes some terrible problems for a lot of people, like bloating, gas, cramps, etc. And the reason that that happens is because that lactose sits inside the small intestine. The bacteria within the intestine eat that lactose, and they can break it down. So they produce gas, they give off all this nasty stuff, and that actually sits inside the human intestine. So that's what causes discomfort in people that can't break it down. So it's really the bacteria's fault. So we know that we can digest milk when we were infants, but what happened in the meantime? Why can't we digest milk now as adults? If we look at that picture, we can see that that is an unreplicated chromosome, okay, because it's a single structure. So again, a little review in there. We all carry the gene for lactase. When we were infants, we know that that gene is turned on or active, and we can all drink milk. So we can drink it, we can break down the lactose, no problems there. However, as adults, it's different. In some of us, what happens is the gene is turned on, it continues to be active, and it produces lactase into adulthood. So again, in some of us that's true, but in the majority of the populations worldwide, that gene is still turned off or inactive. So what happens here to those people that have the lactase gene that still stays active into adulthood? Well, let's look at some examples here. So kind of a little bit of history here. About 10,000 years ago, all human populations were lactose intolerant, so we could not break the lactose sugar down into the more simple sugars. We all carried the gene to produce lactase as adults, but this gene was naturally turned off after weaning, after we stopped drinking milk as infants. So no one could digest milk as adults. Then, and this is evidence that scientists have used or are using to support the idea of how or why current populations, some of us are lactose tolerant. Around 7,500 to 9,000 years ago, a lot of things changed. So what happened is humans began to kind of herd cattle. Many of us became better at farming, and so we kind of learned some things here. And cattle were domesticated in Africa and the Middle East, but this only happened in a few cultures, mostly in Europe and Africa. So in these populations, if these individuals that could herd these cattle were able to digest milk, that was a huge advantage to them. They could digest milk, they could get nutrients from the milk, and that would help them in times of famine, so they would have an alternate food source. The milk was clean, if they didn't have to worry about drinking dirty water because the milk was processed through the animal's body. So this had a lot of advantages for them. So this is kind of introducing the idea of natural selection. Any mutation that happened to have the effect that kept the lactase gene turned on into adulthood was a favorable thing that happened, and that also increased in frequency. So basically what I'm saying is if these people could digest lactose and that was a favorable trait, then hopefully they could survive and reproduce and they could pass that trait down. So scientists believe that based on this evidence, one mutation happened in European populations and several mutations happened initially in African populations. So if we look at this next slide here, we see that there are different things kind of across the world that we're looking at. And basically what we're going to talk about is how lactose tolerance or persistence kind of came about. And there's a couple different ways. So we're going to look at evolutionary history here. Those of us that have deep evolutionary history traces to West Africa, so if we're looking at the West African side of things, or Asia or indigenous American populations, so basically anywhere that there is a 1 on this map here, we are likely to have the ancestral version of the gene that controls when lactase is turned on and off. And this gene is located on human chromosome 2, and the ancestral version keeps the lactase gene turned off in adulthood and actually causes lactose intolerance. So that kind of seems a little bit strange, but I'll get into uh, where lactose tolerance comes from in a minute here. Now, if we look and we see that those people who have a deep evolutionary history that traces back to Northern Europe, which is really more common for us, so many people in America have ancestors that came over from Northern Europe, so we're looking at number 2 there on the map. The C nucleotide was replaced by a T nucleotide. So the point here is that there was a mutation that caused the lactase enzyme to be active into adulthood, which caused lactose tolerance. So many people in America are lactose tolerant because we have ancestors from that area there. Another mutation that could cause lactose tolerance would be found in one of 3 single nucleotide changes, and that would be found if you look at number 3 on the map. That would be found in East African populations. So again, that would be a substitution mutation that caused the lactase enzyme to remain active into adulthood, so we could break down the different lactose sugars, okay? So let's do a quick recap. If you are lactose tolerant, based on the evidence that scientists have, you probably inherited a mutation from your ancestors that allows you to have an active lactase enzyme in adulthood, meaning that you can break down the lactose sugar into more simple sugars. So you then would be lactose persistent. If you did not inherit a genetic mutation which kept your lactase gene active in adulthood, you cannot break down the glucose, which means the glucose will you cannot break down the lactose. The lactose will sit in your small intestine if you drink milk or drink dairy or have dairy products, and so the lactose will not be broken down, meaning that you are lactose intolerant, okay? So there's basically 2 options there, and it's kind of a sliding scale. You might be slightly lactose sensitive depending on how active or how inactive the gene is, okay? So it's kind of like a dimmer switch there. So you need to ask yourself at this point, can I discuss this equation for a lactose breakdown? Do I understand it? Do I understand how lactose tolerance or lactose persistence came about? And if you're not quite comfortable with it, go back through and flip through the slides and ask me or ask someone else if you have any questions. Okay, thanks and have a great day.